Welcome to the second part of the morning session of today. Our speaker is Thomas Simonson. The pod is yours.
Is that better now? Okay. I don't know if I've got a working mic, but at least I don't have an echo. Walter, thank you so much, and all the people doing the logistics. It's great to be in Genoa. It was also really great to have the online seminar series. That was so great during the pandemic to be able to meet people. So after Maria's beautiful neurobiology, I'm going to talk about a bit more methodology, a bit more less fancy systems, but I am going to talk about engineering enzymes, which is an exciting topic. And I'll be using kind of two systems to illustrate two broad approaches. And the first system is an enzyme, the methionine gamma lyase, or MGL, which breaks down methionine. And it turns out that's actually very harmful for a lot of cancer cells. It's been well established and pretty harmless for healthy cells. So that has made this protein a candidate as a therapeutic protein for a while now. It's a protein that uses a cofactor called PLP here, pyridoxal 5' phosphate, which is actually vitamin B6, and which operates, well, first of all, it's a cofactor in over 160 separate enzyme reactions. So it's an interesting cofactor to spend a little time with. It operates here in a form that's covalently attached to the enzyme, and that participates in the reaction, breaking down methionine into these smaller product molecules. The protein's a homotetramer, which you can see over here with 4 active sites. And if we want to use this as a therapeutic protein, there's some work to be done. The first kind of thing is to understand a little bit the details of the active site, the reaction, the structure, the fluctuations. And then the other thing is to be able to engineer it to improve properties, maybe specificity, so it'll degrade only methionine, won't go and degrade your cysteine pool, things like that. There are a bunch of things that crystal structures don't really tell us, and which are nevertheless important for structure and function and for attacking an engineering problem. One of them, which is of interest to us here, in particular, is the protonation states in the binding site. In particular, when you have a PLP cofactor, which has a number of important substates. So it has 4 major protonation states. It has the phosphate group attached here, which can, of course, be fully deprotonated for a -2 charge or singly protonated for a -1 charge. And then it has a connection to the protein over here through a Schiff-based linkage, which likes to be either in this enol form or a keto form over here. So the keto form is zwitterionic in this region, and the enol form is neutral. And so we have these denominations for these 4 substates. So one kind of side product of anything in this area is to actually parameterize this cofactor, and that's a kind of a good thing to have in hand. So we parameterize these 4 forms plus a couple of non-covalent forms using relatively standard ideas I won't go into. And so, the first thing we looked at was, can we guess the PLP protonation state just by looking at structures, right? Okay, so the answer is no, but it's quite useful to mine the PDB and look at the different structural variables and what they do in different situations. So without going into a lot of detail, we've kind of mined the PDB for MGL structures. Oh, by the way, this is a protein that's not present in humans. that's present in bacteria. So we're interested in targeting a bacterial protein for to administer into a human patient. Okay, so we mine the PDB. There are a number of structures in there, and we pull out a number of features, as Maria would describe them, structural features that involve variables in or around the PLP cofactor, things like this dihedral angle, which is here and also here. Okay, these are structures and various structural features vertically, or the N oxygen distance over here, which is also here. Okay, and there's a stereo view of the structure, one of the structures here. And if you look at these variables, or you go in and inspect PDB structures, as fairly common, you cannot guess the protonation state from looking at the structures, because there are multiple different protonation states will be perfectly able to make nice hydrogen bondings, flipping a hydroxyl one way or the other. And you really just can't pull out a protonation, the protonation states from looking at the PDB structures. So then we go into a computational approach, to predict protonation states. And, the method we don't hear about as much as some of the other methods is the one showed here, which is simply to run MD with different protonation states and observe which protonation states best match the PDB. Even if the PDB isn't giving you the explicit proton variables, it gives you these related variables. And there's a good chance that different protonation states will impact these other variables in different ways. So again, I won't go into detail, but the idea is simply to simulate different protonation states here and track, features that we humans have, decided to look at, and that do things that are somewhat interesting as a function of the protonation state. And the result is that, yes, indeed, there are protonation states, there are models, N2 in particular, and Z2 especially, that match the PDB structures distinctly better. Okay, so we're predicting the dominant protonation states, or at least we're inferring the dominant protonation states. There's a difficulty. There are a number of difficulties. I won't dwell on this, but one of the difficulties is the PDB structures, of course, can have different ligands. They can have a bound ion. They can have different things going on in the active site, and you have to kind of deconvolute those from, how those are impacting these structural variables. Again, I won't go into that, but for example, you have to distinguish PDB structures that have an amino acid analog or an ion from those that don't. Things like that. It's not, just a push button. There's some human expertise. Of course, if we want a quantitative picture, we should go to a more sophisticated method. And now we're going to run so-called alchemical free energy perturbation calculations, FEP for short. And I won't, recall a whole lot of methodology, but many of you know, other than the youngest folks in the audience, of course, the idea is to reversibly change the protonation state over the course of a series of MD simulations. We're talking about MD with explicit solvent and molecular mechanics, atomistic molecular mechanics. And so what one typically does is to reversibly change the protonation state. So over here, I'm changing the phosphate from a -1 charge to a -2 charge. I'm doing it in the protein complex. And I'm using as a reference the PLP, the unbound PLP in solution. And, luckily for us, the unbound PLP in solution has been pretty well characterized experimentally. And there's a known pKa value for a close analog, which looks pretty much like this here, with a known pKa of about 6.3. And remember, this kind of phosphate group is fairly typical. The pKa is a little close to neutrality for comfort, for making a guess in an MD simulation or an engineering effort. It's a little too close for comfort. You do need to kind of pin down what is the phosphate state before you go in and spend, postdoc hours on engineering, let's say, let alone experimental verifications. So this kind of technology goes back to the 80s. It's very well known, very useful. There are a number of difficulties, well-known difficulties. One is that it's hard to, sample sufficiently the fluctuations of the system because there's slow variables in there, and if you change an ionic group, it can take a long time for the MD to adjust to that. So this is just a kind of view I won't go into much, but just to show that we run a lot of sampling and we do a lot of kind of human inspection of what's going on in the simulations to determine if our simulations, track pretty well what we've seen in our 5 microseconds of plain MD. And it will be, I think, interesting in the future to put together some more automated methods, kind of, in the spirit of the machine learning that Maria talked about, to track sampling and convergence and things like that for these relatively large data sets. There is a window here. We won't be doing that, but if it interests somebody, I'm pretty sure there's a kind of window here of useful machine learning work to do, kind of monitor certain types of free energy simulation. So one difficulty is sampling, and that's a lot easier nowadays than a few decades ago. It's still an issue. Okay, now something more related to electrostatics, for this audience. There's a difficulty people became aware of, only maybe a dozen years ago, much more recently. Kind of a technical difficulty with the PME simulations we use, most commonly for this type of simulation. So here again, you have a protonation or deprotonation reaction going on in the protein in a box of explicit solvent. Down here, you have a much smaller model compound. And, the thing with the potential particle mesh Ewald in its normal implementations in the most commonly used software is that they impose what we call tinfoil boundary conditions. And what that means is the simulation box is electrically grounded. That's a convenient setup of the MD technology. We actually have simulation boxes that are electrically grounded. And what that means is that the electrostatic potential averaged over the box is 0 at all times. And normally that's not something you need to think about a whole lot when you're just running MD, or you're doing, even if you pull an ion through a channel, do a PMF. That has no effect whatsoever, the fact that the box is grounded. But when you start to compare a big molecule and a small molecule, you do have to pay a little bit of attention. Down here, the box is mostly water. If the box is grounded, it means that the electrostatic potential in the solvent region of the box is essentially 0, because the box is essentially all water. However, if you're saving money up here by running a kind of smallish simulation box, up here, if you run a huge simulation box, it's mostly water. The electrostatic potential is 0 throughout the box, including the bulk solvent region. And so these two systems are comparable, and you can forget, you can, stop listening. But usually we're trying to save money by kind of, using a smaller box that kind of we can just get away with. In that case, the electrostatic potential in the bulk solvent region of the box cannot be 0. Okay, so you're running two systems they're not electrically consistent, and now you're going to go put a charge into both of these boxes. Okay, but there's an electrostatic offset between the boxes. So you need to compute that box, that potential offset between this part of this box and this one is 0. You need to compute the potential here, offset it so it matches this one, and multiply by the inserted charge. Okay, when you do that, you're going to get a kcal correction. which will typically be about the same size as everything the FEP is giving you. You know, the delta delta G is probably about 1, and this correction is about 1. So it's something to, you know, at least be aware of when you're doing ionic transformations with FEP. That's the second point. There's another one, which is, you know, this is now really for the young people in the audience. We're in a situation here where we can't pull out the phosphate pKa by just comparing one of the -1 states and one of the -2 states, because there are multiple -1 states and there are multiple -2 states. Okay, here there are only 2 of each, but you know, in a general situation, you have, if you have more than one, you need to be pay a little bit of attention. Of course, you know, the pKa shift of my group of interest in the protein relative to my model compound, you know, can be related to these standard deprotonation free energies, which I can obtain from FEP up to a constant that will drop out. Okay, but this deprotonation free energy is not like the N1 to N2 FEP result. It's a Boltzmann average over the -1 states and a Boltzmann average over the -2 states. And so you do actually need to kind of, if you're going to simulate, for example, the N1 to N2 transformation I showed you a moment ago, it's not enough to give you the standard deprotonation free energy. You need to take into account the other states, E1 and 2. And so you have this, you know, slightly more complicated multi-conformation formalism, you know, which will be obvious to your supervisors, but, you know, you need to be careful and realize that there's a slight issue. And if you're a PI, you know, then, you know, you're not going to have problem writing down a multi-conformation formalism, but you just have to be aware that, you know, your postdoc or student might not have realized that, you know, it's an issue. So we studied all these 4 states and their transformations in the protein in solution with and without the methionine substrate. And just a couple of interesting things. First of all, here we have the pKa shift. So for the model compound in solution, this is our reference. It's a value of 0. The protein downshifts the pKa, so it really stabilizes the -2 phosphate significantly. Excuse me? These are pKa units here, or pH units, if you will. And then these are percentages of the populations of these different substates or tautomers, if you will, in solution. The protein apo means there's no methionine substrate, and then with methionine down here. And so the protein shifts the phosphate more strongly to the -2 state, even more so than the model compound. I'm happy to be interrupted, no problem. My talk is a little short, so I have plenty of time for discussion, actually. Well, from the relative free energies. Okay, there's a little bit of algebra, but there's no physics. It's just, you know. Well, we did all the relative free energies of these four states in all these 4, you know, situations. Yes. I didn't show you the free energies. I'm just showing you the populations. Okay. But yeah, you have, you know, the kind of the substates related to the overall free energy. You have this conformational correction, you know, which can be purely entropic in some cases, and so on. Anyway, the point is, the protein likes the -2 phosphate. And there's a kind of interesting small effect that goes on when you add the methionine. That's the second point I want to make here. That so N2 and Z2 are predominant. In this case, it's mostly Z2 without methionine. And then when one binds methionine actually pushes the system from the let's see, from the keto tautomer into the eno tautomer. And what's interesting about that is the reaction can proceed much better from the N2 substate. The structure in the active site is such that the N2 really helps promote the reaction. So methionine binds, doesn't really change the protonation state, but it pushes the PLP into the tautomer that favors the reaction of methionine itself. So there's a little bit of substrate-assisted reaction going on, which is kind of neat. Okay, so at this point, we know enough about the system. We feel we could go on and do some engineering. But there's another difficulty now, which is the kind of complexity of the overall reaction. So just a moment here to look at the different reaction steps, because you have the methionine substrate, the enzyme, the PLP, and these guys, M and P, have to bind in some order, PLP first, or methionine first. At some point, they could both bind. And the colons indicate non-covalent binding. Okay, but then the PLP needs to become covalently attached. Actually, we know experimentally if it's not covalently attached, the reaction won't proceed negligibly. Negligible activity. So at some point, the system has to move from this box over to this box where PLP has a covalent linkage, which is marked by a dash. And it can occur, without methionine, and methionine binds second, or vice versa. So there are a lot of different kind of intermediate states, and it's not obvious when you start out which ones should we be engineering. What property exactly should we be engineering? So there's a little bit of simple analysis one can do, and it's just one slide, don't worry. And then from over here, you of course have to go through this state, which is effectively a Michaelis complex. And now the methionine can break down. The actual reaction can occur. But you do have to go through this state. So the states in boxes involve non-covalent binding, unbinding. And then the red arrows invite making or breaking covalent links, right? So if we make the assumption that the non-covalent steps are fast and the covalent steps are slow, then we get a steady state, just like Michaelis-Menten theory. You get a steady state means that the population in this red box is stable at the steady state, just like in Michaelis-Menten that we all teach, right? And if we assume that the covalent reactions are irreversible, which is pushing a little bit more, but still, probably okay qualitatively, then we get this simple result down here that the formation of products, can be written like this. And then the steady state assumption tells us that we can write it in this form over here. And so the activity level is really going to be tuned by the amount of non-covalent PLP binding. So that's a good engineering target. Okay, so we could decide to engineer the protein to increase non-covalent PLP binding. And if we do that, we have a good chance of directly impacting the activity. Okay, so it turns out engineering non-covalent ligand binding is something we like to do. It seems a lot easier than engineering covalent binding. To me, it's certainly technically from a computational point of view, but we haven't yet really done it for this enzyme. So I'm going to move now to another approach. This was all atom, explicit solvent, MD, free energy simulations, and so on. And we could push ahead with this method and engineer the binding site. But we're not doing that right now. We want to move to a high throughput approach. And so now I'm going to say a little bit about a high throughput approach for computational enzyme design. Okay. And I'm going to describe it using a different system, which we've actually done. But then we're in a position to do the same thing for MGL. Okay, so computational enzyme design is a subset of computational protein design. So when you say computational protein design, you've moved to a slightly different level of theory, software packages, and so on. And that's what I'm going to talk about now. So computational enzyme design in this CPD world typically will choose just a few amino acid sites, a few amino acids in the active site to actually and actually let them mutate freely in search of some property of interest. Okay, and so the property of interest or the design criterion or criteria could be substrate binding affinity, could be transition state binding affinity, could be binding specificity of one or the other, or some combination of these. Okay, now it turns out designing for binding, substrate, transition state, whichever, is actually a hard problem in this protein design universe, because you need to mutate your active site in such a way that you increase the stability of the holo system, but you decrease the stability of the apo system. And you need to do this with a single mutation. Each mutation has to sort of try to do both of these things. You've got a positive design problem and a negative design problem, and you're running them, they're intimately coupled. Turns out that's actually hard. And if you, work at it a little bit, you will find that it's hard, and people know that it's hard. So there are a couple of approaches to attack this problem. By far the best and most efficient is based on ideas from Wang and Lando of a few decades ago, which are closely related to the ideas of metadynamics. And it's basically methods for importance sampling, sort of clever importance sampling, which we borrowed and repurposed for this particular type of problem. So I'm going to tell you a little bit about Wang-Lando adaptive Monte Carlo. And these are colleagues who did this work in the 80s, I believe. This can be used in an MD framework, and then it changes names. It becomes metadynamics. It was first applied to protein design by a Swedish colleague 10 years ago, who did a kind of a feasibility study, but never did any actual application. So we rediscovered it. We literally rediscovered it. Wasn't aware it had been published. We were very disappointed to find that somebody else had invented it. But okay, so be it. And there's a related line that's been developed in Charlie Brooks's lab using so-called lambda dynamics, so using MD, not MC. But the basic idea, it doesn't matter what level of theory or what type of simulation you're using. It basically is to adaptively flatten the free energy landscape of one state and then sample the other state with the flattening bias. That's a very generic idea, which turns out is really neat. And I will go slightly into detail, but to summarize, if we had flattened the free energy of the apo state, it means the protein without our ligand, okay? And we do this by adaptively learning a bias function. So if you think of, earlier we saw a PMF for a hydronium working through grammacidin, there was a free energy profile for the ion moving through grammacidin. So if you subtract that free energy from your system, now you have a flat profile. You've biased and flattened. You didn't do it adaptively, you did it post hoc. But in practice, it works very well to do it adaptively. Once you've done that, you can add your ligand, simulate with the same bias. The bias subtracts out the apo state. And now you have a simulation that's controlled by the binding free energy, literally controlled by the binding free energy. It's almost like magic. So here's an example. It's not a toy example. It's a real example with a simple situation of just 3 mutating side chains, but it could be 4, 5. So these are side chains in the enzyme I'm going to talk about in a minute in the active site. And this is the apo state. And I'm showing you the simulation results in the form of a logo. So we have 3 active site positions we're allowing to mutate freely over the course of a Monte Carlo simulation. The Monte Carlo approach is literally MCCE with mutations, okay? So we have our own Monte Carlo package, It's the same technology Marilyn described earlier, except we have side chain mutations as well. Okay, so we're doing Monte Carlo in a space where side chains can mutate and they can rotate around and explore rotamers. And when you do that with this protein in the apo state, these three positions occupy different side chain types over the course of your simulation. You can represent that as a logo. Okay, so there are some dominant types. Now we adaptively learn a flattening bias, and we very effectively flatten the sequence space, because now you can see each of these positions, basically occupies all side chain types with equal probabilities, right? Now we add the ligand. Okay, so because our bias has flattened this guy, it means the bias is the free energy with its sign changed as a function of sequence, mind you. We add the ligand, and now we get these populations here. And in this particular case, it's kind of neat because the wild type sequence is LAI. And now the sequences are literally populated according to their binding free energies. And this is Wang and Lando, it's not us. We've just repurposed this into the Monte Carlo world, where we mutate side chains over the course of a Monte Carlo simulation, and we do it in such a way that we get Boltzmann statistics. We're not doing simulated annealing, we're doing Boltzmann sampling. That's why it works. So the adaptive flattening is, this is a well-known idea, but I give you a quick breakdown here. Now I have a system here where I have sequences along this axis. These are the different sequences I would like to explore. And here I have a free energy axis. Okay, this is the initial system. There's some sequences that are very favorable, some that are much less favorable. And if I start running Monte Carlo to simulate this sequence space, initially I'm going to be spending time down here in my basin, my well, right? And what I do is every so often I add a little chunk of bias to the current sequence. So I add this little red energy blob wherever I happen to be at that time. And every so often I do this. And so over time, in the early stages, I'm basically adding red stuff to the well, and I'm starting to fill up the well. And then over time, I really aggressively fill it up. And eventually I make a flat free energy, which is just a straight line. And when I reach that point, as you can easily see, the bias is just the free energy turned upside down, right? So, the bias to some level of approximation, which is a practical decision you have to make, is literally the apo free energy with its signs changed. So now you simulate the bound state, including the bias function, and now you have a simulation that's actually controlled by the binding free energy and populated according to the exponential of the binding free energy. It's just like magic. That's really, and then you can do many other things with this, believe me. You can make variations of this idea. You can make a dozen variations. Many of them are extremely useful. So I have no idea how. Perfect. So the actual level of theory here, molecular mechanics, we have a continuum dielectric solvent, which is GB plus a hydrophobic term. The hydrophobic term is worked on the work of my friend Themis over here. He's the L. L here is Lazaridis, and K is Karplus. We have a conformational space with a fixed backbone and discrete rotamers. We can move to a flexible some backbone flexibility. I won't describe it. We have replicate exchange Monte Carlo, where the moves include mutations. We have a software package that's freely available, a lot of publications. I'm going to skip over this because, so this is the system, one of the systems we've most worked on, methionine tRNA synthetase, and this is a huge issue for genetic code expansion. Okay, these synthetases are targets for engineering for genetic code expansion. So you want to engineer a synthetase to modify the association between a tRNA and its anticodon at one end and the attached amino acid at the other. If you do that, you can scramble the genetic code, extend it, make it degenerate, change codon meanings. And all this has been done for about 15 years now. We looked at the methionine enzyme. We looked at these, target amino acids. And I will skip this. This is some validation, reproducing some published data very nicely. And so the main test we did was to look at redesign the enzyme for the natural substrate activity. So make new mutants that remain active. Okay, so you're redesigning not for a new amino acid, but for the methionine activity. Our design criterion was transition state binding, not ground state binding. So we have some quantum calculations go on to make a transition state model. It's quite simple in this case because the reaction mechanism is very simple and well known. So we have a molecular mechanics model of the transition state, and we designed for binding. We had a great situation where we actually had experimental collaborators who were motivated. It's so hard to achieve in our field. Sometimes you wait years and years to get experiments, done. It's better to do them yourself. My advice: set up a hybrid lab if you're young, because anyway. So we had blind tests where we took 40 top predictions. We picked 17 for experimental testing. All 17 were active. None of them, succeeded in doing better than the wild type. We predicted a few activity gains. Those were within our error bar. We did have detectable activity. We never got a better result than the wild type. These are log catalytic efficiencies, computational, experimentally, and so on. And then to wrap up, we're very interested in expanding the genetic code towards amino acids that have non-standard backbones. Non-standard side chains, there 200 to 300 have been introduced into expanded genetic codes, somewhere between 200 and 300, because I don't keep up on a monthly basis. It's maybe 250 or so. But nobody has succeeded, other than some of our earlier work, in engineering a synthetase to prefer an amino acid with a non-natural backbone. So we were successful with D-tyrosine, to get D-tyrosine activity into one of these. And recently we worked on beta-methionine. Now, if you can add an additional methylene to the polypeptide backbone in a controlled way, and you think about it for a while, that would open up enormous possibilities for, engineering structure, just the way if you can insert a D amino acid in a controlled way into the active site you're trying to engineer, that can make the difference between success or failure in your enzyme engineering. Okay, so adding a beta methylene to the genetic code, a backbone methylene, that's a challenging thing to do. So we were able to get variants that had a reduced preference for alpha met by up to a factor of 30. So it's a kind of semi-success. I would say it's a success because these enzymes are 3 billion years old. They're among the first enzymes that emerged, and they're not easy to modify, in a way like this. Okay, so with that, I will stop, just acknowledge several really, really excellent students and postdocs, and the blue ones who did most of the work here. Advertise, do my job with respect to Springer. I should advertise this because, Springer made the effort to publish it. And this is our software, which is complex but well documented. Thank you all.
I think we have time only for a couple of questions.
So this is kind of a comment and actually a little to Miguel's point. So when you look at protonation states in the PDB files, we actually found that there were almost 10 times as many of it. So one thing is, do you find multiple protonation states, all of similar energy? And this is a comment that I think in the PDB, you will see more protonation states at equilibrium if you don't do molecular dynamics. Once you start doing molecular dynamics, then you move into defined protonation states that are many fewer than in the original file. Because I think the original file is probably has the background of lots of different positions for the protons.
When we sort of mine the PDB for structural features, we don't make any assumptions about protonation states. We're mining, the protonation states are like a hidden variable, which we're trying to infer. So we're mining, other features, and from that infer a protonation state, we use the MD simulations. And of course, you're right. The reality is more complex than our 4 MD models because surrounding groups, are fluctuating a little bit. This is true. There's plenty of complexity. Jana.
Hi, Tom. This is a very nice talk, beautiful talk. So to your protein engineering part, did you try empirical approach, such as the David Baker's empirical energy function?
Well, yeah, good question. I mean, we tend to benchmark our model against Rosetta for typically for whole protein design. So that's kind of one of the standard benchmarks in this field is to do whole protein design and look if you get a native sequence recovery, look at the similarity to PFAM. And so when we do that, those benchmarks, we compare to Rosetta. Protonation states, we also do pKa benchmarks, and we do them with Rosetta as well.
For pKa, you can easily run a constant pH. Have you tried that?
For sure. Well, these simulations can be viewed as constant pH Monte Carlo. When we do pKa, we're running constant pH Monte Carlo and pulling out pKa's that way.
No, I was referring to your the early part of your talk where you tried a bunch of combinations, right? Instead, you could run a constant pH.
That's right. And that's great technology. And if we can run explicit solvent with the same amount of sampling, with that would be probably a better alternative, than the kind of alchemical state-to-state connections. Yes, that's
will be a lot faster.
Thanks again, our speaker.